Thank you for joining us. I hope that everybody is in good health and that you're remaining safe and vigilant and practicing social distancing during these trying times. And I would like to take a moment to say thank you to all of the sponsors that you see on your screen here, who are the folks that are responsible for allowing these learning opportunities to continue. I'd also like to thank ALAS for allowing us to socialize these events on the ACE community, and specifically John Farrar, who's the Director of Business Development and Communications at ALAS, for all of his guidance and support as we've set up these webinars. Please note, however, that although ALAS has helped put these together, they are not sponsored by ALAS, and ALAS does not constitute endorsement of any of the companies or products being discussed. So that goes for us and everybody else who is participating in these events. The webinars are provided by the individual organizations that have all stepped up during these unprecedented times and have agreed to provide these presentations. I hope that you've been learning a lot from them. I personally have been really excited to see some of the things that have been going on. Continue to check the ACE community postings and the ALAS LinkedIn group for any additional upcoming webinar details. I'll note at the end what's going on next, but I think probably most of you hopefully are aware that there won't be any webinars next week because it will be the opening for ALAS, the national meeting, and hopefully everybody will attend that. For today's webinar, everybody is muted. So if you would like to ask a question, you'll need to do that through the chat screen and in the bottom of your screen. If you have any trouble with that, well, if you have any trouble with that, you can try to reach out to Shelly, who helped get you the details for this event, and she'll keep an eye on things coming through through email in case that's needed. All right, when we do get to the end, I will address all of the questions as possible, and if I don't get to your question, we will do a follow-up for you after the fact. In addition, this webinar is being recorded, and we'll provide a link to the recording to everybody that attended so that you can refer back to it after the fact or share it with others if you'd like to do that. So with that, I'm going to jump into today's topic, automated sampling in a pandemic. Okay, so the more official title than what I just said is Automated Sampling for Safe, Effective Drug Development Studies During a Pandemic. What we are going to talk about today all revolves around the Culex automated blood sampling system, because that is the one that, that we are most familiar with here. But honestly, this is true for any kind of automation that you can put in place. We will give you an overview of what the Culex is so that you can better understand that. The Culex system is a system that lets you stay calm, cool, and collected. It lets you sample from awake and relaxed animals so that you can uncover data that stress could potentially hide. Everything is collected into a refrigerated collection system, and that allows you to collect any time of the day or night. And you know that everything is being collected. You can have complete confidence in that sample collection because there is both a detection built into the system as well as a notification system to let you know what's happening throughout the course of the study. So what we'll do overall today is give you first an introduction to the Culex, what the systems are and what they can do. We'll talk about the impact of automation on study design. We'll talk about the impact of automation on efficiency in the lab. And we'll talk about the impact on safety. And then you will get to hear me be quiet for a little bit and Mr. Andy Zuvich speak up and share with you a case study of how they've used the Culex system for just these things. All right, so what is the Culex? So at a basic level, we're going to start with system components. The first thing is that there's software to program the volumes and times of blood that you would like collected. The controller is the unit that directs the flow of the blood, keeps it all contained. The collector is filled with treated vials and is chilled, and all of the samples are delivered into that system. That whole piece integrates with a tether or a cage, and there are different options to that, which we'll look at momentarily. There's also a cart or a platform for housing the system, so that just makes sure that it is all stable and spaced properly in some cases. And then finally, there's a tubing set, which helps move and contain the blood. All right, so much easier to show that video than it is to try and explain everything, but it should have given you the basic overview of how everything is collected. You have that understanding now, so that will let us move ahead. So what you saw there is the rat system, but as I mentioned, we have sampling capabilities for a variety of animals and a variety of setups. So starting from the left side of your screen, you'll see the rodent stations. And there are kind of two primary options available. The first is a swivel-based system that works with a home cage setup. So that is really when you want to do just simple, straightforward blood collection for rats, mice, and other rodents. The second option is a rat turn-based system. So the rat turn is a responsive caging system that replaces a swivel or replaces a commutator. We have a lot of other webinars and information available if you'd like to learn more about that, but the basics are that it allows you to have multiple types of lines connected to your animal so that you could do something like blood collection, infusion, bile collection, and bile salts infusion all at the same time in the same animal because you don't have any swivel channels to worry about. And so those systems, as far as the blood sampling go, work exactly the same. It's just the way that the animal is housed and contained and then what you can do with them on study. As we move to the right side of the page, these are our large animal systems. So the first one you see is our CoolXL. So everything works exactly the same, but these large animal systems are built to accommodate larger volumes because you're going to have larger blood volumes that you collect and larger connected volumes between the instrument and the animal because of the size of the caging. These systems have been used for pigs, for primates, for dogs. They could be used for other things. Basically, as long as you can put a catheter into the animal, you should be able to collect samples from it. And then the final picture is a caging system for large animals, specifically for mini pig research models, which is called the pig turn. And this incorporates with the automated sampling system so that you can, just like with the rat turn, do multiple types of samples from multiple animals. So why would you think about automating? The first reason that many folks consider this is because it improves staff and scheduling efficiency. We're going to go into greater detail in some of these things, but that's one major reason. Another is staff safety. This is especially true for large animal models, but there are reasons across the board. It also lets you do low stress sample collection. So that is primarily focused on the animal. The animals are under less stress, but that also impacts the researchers doing the work. And then there's a lot of flexibility in the types of data collection. So one of these key areas, or multiple of these key areas, are the reasons why most of our customers bring automation into their lab. But really what it comes down to in the end is that we are all trying to do more with less. So whether that is less compound, and so you're able to save it through some of the dosing mechanisms in the system, whether that's because you have fewer people, whether that's because you have fewer people because some of them are working from home, or another way of looking at it is that some of the ways that you can combine studies on this system let you use fewer animals overall, and so you're able to collect more data and use less animals to get that data. So we're going to dig in a little bit on some of these so you can better understand the impact of the system on those parameters. So we'll start with the impact on study design. So we know that sampling method influences stress hormone release, but here is a good example of it. So this is a little bit complicated. So bear with me for a second while I explain it. But this was done by a group that was looking to do safety pharmacology studies on their Culex automated blood sampling system. And they wanted to make sure that when they did that, they weren't going to introduce any outside parameters that would affect the animal stress. So they were doing PK alongside of telemetry for very stress sensitive parameters like heart rate, blood pressure, ECG, and did a large validation study to see what was the difference between the way they were doing it before and the way that they wanted to do it going forward. So you've got a couple of parameters here. The first one is the tail bleed. That's the gray box with the lines through it. And that's a pretty straightforward, common way to collect blood in animals. We all know that's going to be relatively stressful. So this one isn't a big surprise. The animal is put in a restrainer, usually warmed up to vasodilate, and then a needle stick or a clip will take the blood sample from the tail. The yellow is ABST. That was this group's terminology for automated blood sampling plus telemetry. So they were using the Culex for this system. And the gray is home cage. So in that scenario, the animals were in a home cage with a harness and tether and swivel, and the animals were not handled during the blood sampling, but people were in the room. They were there at the location where the instrument was, and so no touching the animal, but just being present. And what they found was that for for corticosterone, insulin, and prolactin, several of these stress hormones, not only do you see what you might expect, that the levels of stress hormone were lower for the hands-off methods versus the hands-on, but also there was a statistically significant difference between automated completely and the methodology with humans in the room. So just the humans being present, even if they didn't touch the animals, could have an impact on stress hormones. We've also seen similar responses in primates. So in this example, you're looking at a heart rate in primates on an automated collection system versus manual. So this is the automated manual is referring to blood collections, but we were also taking heart rate during this period. Those orange arrows, both of them are times when humans had to come into the room and do some more invasive measurements or touch the animals in some way. So you can see a bump up at the beginning when everybody got dosed, and you can see that bump a little bit later when they all got their checks. But you can see here that there is a difference pretty easily observable between the heart rate in an automated sample collection group and the heart rate in the manually touched group. One other note I'll make here is that this study went on for a 24, 25-hour period. So we'll come back to that in a moment. So some people tell us, I don't really care, right? I'm not collecting that kind of data. I don't care about heart rate. I don't care about stress hormones, and that's fine. I mean, everybody has a different goal in mind, and sometimes that's not going to be such a big deal depending on your study goals. However, it's not just those things that's impacted. The way that we interfere with animals, the way that we design our studies can have pretty significant impacts on the results that we do care about in a lot of cases here, which is the pharmacokinetics. So this study took a look at automated versus manual dosing. Everybody was collected with automated blood collection in this case, but the difference was that either they were delivered a dose through a gastric catheter directly into the stomach without touching the animal, or they were gavage dosed through the standard method, handling the animal and putting the gavage needle into the stomach. And you can see that there is a difference between the automated dosing and manual dosing. So again, that method of intervention with the animal is what makes that impact. The idea here is that when the animal is stressed out and goes into fight or flight response mode, the blood is shunted away from the digestive tract out to the extremities, so it's ready to do what it needs to do. And when that happens, your blood that's gone away from the digestive tract, where your drug absorption is starting its process, is impacted. So less blood in the digestive tract means it's a little slower to get into the system. It might not get in at the same amounts. And this is, in some ways, if you are looking at it from a perspective of this is the way we've always done it, and you want your data to stay the same, then that's something that we hear from time to time. But I would argue against that when you're doing that, you're stressing the animal out in a way that you're not likely to do to your human models later on. And what we're trying to do is have the best data early on that is translatable to the human models later on. And we don't tend to take people by the back of the neck and stick a gavage needle down their throat to give them a dose. People can understand and are able to take that medicine and are able to understand that it might have a positive impact on them. And so that taking of medicine is not an actual stressful event. So as much as possible, it's a good idea to try to mimic what we're doing later with humans as early as possible with those animals. And this is just a point to say that it is not only in rats or rodent models that we see this. There is a similar impact across species. This just happens to be one in primates, where again, you can see that difference between what happens when we do automation versus manual hands-on collections. So what this lets you do overall is get better data. So you're able to uncover data that isn't possible with manual methods. We can see those stress hormone changes. We can see the heart rate changes that would be masked by humans being around. And you're able to reduce the animal stress and all of those associated complications, whether it's the actual stress hormone release or whether it's something more like the changes in the absorption of the drug, like you saw with the dosing methodology. And then the final thing is that it lets you take samples any time of the day or night. And that might seem like more of an efficiency thing, and it is. But the other point here is that rats, at least, are nocturnal. So the time when we tend to take all of our samples from them is their nighttime. And that, again, is not when humans tend to take their medicines. So as much as we can do to kind of keep things similar, the better off we'll be. And this system lets you at least make that choice. You might choose not to do it, but you could if you wanted to. It also lets you combine studies, so you're able to get the full profile of your compound in a single animal. And this has been especially useful for a couple of our customers using the system for a combination of the PK/PD. So you heard that reference with the folks doing the ABST. They're combining PK with the cardiovascular parameters in their safety pharmacology studies. So prior to having this system in place, they would have 8 animals over here for the PK, 8 animals over here for their cardiovascular. You couldn't mix and match because the data would get all skewed when people took their blood samples. So you'd have 16 animals on study and no correlation necessarily between them. So if there was a missed dose or there was some kind of problem, you wouldn't have any way of knowing that, cross-referencing them. So what they were able to do is bring it together and reduce your animal numbers by half. So we just automatically went from 16 down to 8 animals, and you're able to get all of that animal and all of that data in a single animal. That animal becomes its own reference point of the pre-dose and post-dose and what happens on an individual and a group basis. So we've had customers use that kind of methodology for rats, but also more recently in canine models. And the focus there was as much about reducing animal numbers as it was getting results earlier in the process so that you were eliminating costly mistakes in the drug development process later on. You've heard me mention a few things that you can do with this system, and this does depend on your model and the setup, but this is the other important part about it, is that you can do lots of different types of sampling. So I've just talked about the cardiovascular parameters and blood sampling, but there's also additional physiological monitoring options, ECG, EEG. You can integrate optogenetics, fiber photometry, interstitial fluid sampling like microdialysis and open flow microperfusion, and activity data. And a lot of that can be done at the same time in the same animal, so that you're just getting more information earlier in the drug development process to make better decisions. All right, so you have kind of an idea of how automation can impact the data, the results, the study design, but we're going to talk a bit now about efficiency. So just so that we have an example to work from, because everybody runs their studies a little bit differently, I've just laid out the parameters here. We're going to talk about a 12-rat PK study, 24-hour collection period with 10 time points, one of those being an overnight period. So when you're talking about traditional blood collection, there's preparation. So it's just going to be a little different depending on your lab and what you do, but more or less, you're going to have to come up with a timing spreadsheet so that you've staggered the dosing and sampling. You're going to have to prepare the space that you're working in. If you're using a water bath or heating lamps or other kinds of equipment, you'll need to get that ready. If you're using catheters and you don't need that, then that's kind of replaced by preparing your flushing and dosing syringes. And the overall preparation of all of the supplies. So not too bad, usually about maybe an hour and a half to do all of that. Kind of depends on how experienced you are at it. And then we're going to take a look at the same kind of situation for Cool-X. So this also is highly dependent on your experience, but there's a couple of other things that come into play here. So you're going to put tubing sets onto the systems, you're going to connect the animals, load your methods, and most of our customers do some kind of system check just to make sure everything is doing what it's supposed to before it goes on, but that's sometimes not done before study, sometimes just done on a schedule. So it maybe takes a little bit longer, but that really depends. You know, when you first get started, putting a tubing set on can take you 10 minutes, and then once you get good at it, it takes you like 3 minutes. So all of that is a matter of practice and how you get there. But overall, not a huge difference there, right? About an hour and a half for traditional, maybe 2 hours for the automated. Where it really makes an impact is this next section, which is the actual execution. So when you're running the study with traditional blood collection, you're usually doing staggered dosing and staggered collections so that you can make sure you get all those time points because you have to have a person or multiple people there to do it. You're going to have to do some animal prep. So either that's you're warming them or putting them in the restrainer ahead of a tail vein, or it's that you're flushing and locking the catheters after a catheter collection. And then there's also a record keeping component. And we're talking about this full 24-hour study, so 8 to 10 hours is not unreasonable. Some of that is going to be in your overnight collection as well. With Cool-X, there's a pretty stark difference here because you don't do very much hands-on. Once those animals are on, basically all you're going to do is collect pre-dose, which is done with the push of a button. You administer the dose, also can be done with the push of a button if you prefer. And often the extra time there is spent just observing the rats after dose to make sure that you don't have any adverse effects from your compound. Cleanup is another one where it's about the same in terms of amount of time, but the activities that you're doing are a little different. So with traditional blood collection, you're probably doing a little bit more on the actual cleaning, getting rid of your supplies, and document review, because most of that is a manual process. Whereas with the Cool-X, you're probably doing transferring the animals off of the equipment, removing tubing sets, giving everything a wipe down, and there's no time really in transferring the report. If you're networked in your computer, you can have it go right to where it belongs anyway, but otherwise it's as simple as moving a file. All right, so for traditional blood collection, we're talking about probably about 13 hours of total active time. 2 of that is in the overnight period. And depending on how good you are or what your system requirements are at your company, that's going to take 1 to 2 technicians. So I know some folks out there say 12 rats they can handle, no problem, and others would be hard pressed to do 5 or 6 on their own. So that's going to vary from place to place. So if you actually did have to have 2 people on that, then you could be looking at as many as 26 hours to complete that study. And with Cool-X, we have about 5 hours of total active time. You don't have to be there for any overnight samples, and that could very easily be managed by a single person. There's a little bit more to it as well. So in kind of talking with some colleagues and making sure that I thought the numbers that they thought in terms of that rundown, one of my colleagues said an 8-hour study is a 10-hour day. Right, so if you've got an 8-hour PK, you're not there 8 hours. You've got to be there ahead of time to make sure everything is set up, ready to go. You're going to be there after the fact, collecting and processing samples. And so there's definitely truth to that. And the nice thing about Cool-X is that even though that preparation might take you a little bit longer if you're still learning, it can be done often the day in advance. So a lot of times what folks will do is do all of their setup on Monday and then dose on Tuesday morning and be done within a reasonable amount of time. So that's a very nice aspect to be able to space your time out like that. With traditional blood collection, those first few hours of the study are extremely intense. There is no way you're going to any meetings or getting any emails in or doing anything outside of that. But for Cool-X, outside of the dosing, there's relatively little stress on on you throughout the day. You're basically doing that first bit of dosing, keeping an eye on the animals for a minute, and then you come back kind of at your leisure to take samples and process them, which is extremely useful for getting other things done. I kind of touched on it, but when you're doing traditional blood collection, the catheterized animals can be even more time consuming in terms of preparation. This is because if you are not buying pre-filled syringes, you have to fill them yourself. So this is for every animal, you have to have something to pull the lock, to flush the catheter, and to replace the lock. And that takes a lot of time and a lot of energy to get set up. With Cool-X, everything is automatically flushed. So every time the catheter does a draw, everything is flushed, and there is a catheter maintenance patency system called TEND that gives a little pulse of the saline every couple of minutes to make sure the catheter stays open during the non-collection periods. And overall, with traditional blood collection, it's difficult or impossible to get other work done on a study day. You're not likely to get very much done, especially in those morning hours. But with Cool-X, a lot of our clients find that they use their time to run second, shorter studies or to engage in some more exciting activities. And you'll hear a little bit about that in the case study in a bit. So hopefully that helps you understand how this system or automated systems can make your lab more efficient. You're able to reduce the number of staff needed for the manual collection. You're able to redirect technicians toward more rewarding activities or sometimes extra studies, sorry guys. And you're able to take samples anytime without staff present. Okay, so we've talked about the impact on study design, we've talked about the impact on efficiency in the lab and what you can do with your staff, and now we're going to focus on the third important factor, which is the impact on safety. So one really great benefit of a system like this is that you're reducing exposure. So this is, using a system like this leads to a drastic reduction in human and animal interactions. You don't have to move them, you don't have to restrain them, you don't have to do anything. Once they're connected in the system, that's really the only touch point, is the connection and the disconnection. That's it. So a lot less opportunity for bites, scratches. There are some systems that don't use any needles at all, but if there are, it's limited. It's the one-time connection to your vascular access port, and then you never worry about it again. So you're diminishing that interaction and reducing the chance to get needle sticks or bite or scratch injuries. So obviously that matters across all species, but it's a little bit more important for some of our non-human primate studies. The other point here is that the blood collection is fully contained within the tubing. So when you're done with the study, everything comes off. There's no exposure to the fluids. It all goes into your biohazard very quickly and easily. Okay, so we're able to reduce exposure. We're also able to reduce exposure. All right, so we were talking about animals and bites and so forth in the last section, but obviously right now we also have a lot of limitations to how close we can be with other people. So these studies can be run efficiently with limited staff. As I said in that example, a single person could very easily run 12 stations and not have too much trouble with that. You're able to monitor progress from outside of the room. So if you had one person in and one person out, just kind of checking things on the software, on the notification system, then you're able to do that without having to actually go put your eyes on. And it's also reduced the interaction with large species. So in the scenario above, we were focusing a bit on the actual interaction with animals, but a lot of those require two-person protocols. So you need to have one person that's there for just safety purposes. And once you've got this in place, again, there's still that connection and disconnection, but once the animals are connected to the system, there's almost no interaction. So you don't need quite so many people around for those blood collections. And finally, you can reduce exposure. No overnight blood collections mean a couple of different things. One is you don't have to wake up in the middle of the night to drive to work. And so for those of you that have done that, myself included, it's a little bit dicey. Setting your alarm at 3 in the morning when you're not used to getting up at 3 in the morning is not necessarily the safest thing to do. You also don't have to come into the office, depending on your setup, and struggle in dark lighting or red lighting to collect your samples. But the other part of this that we don't think about so much is that whether there's actually a safety risk or not in the area that you're in, coming to the office alone is not really a great idea, whether it's because of safety risks when you're in the building or whether it's because there are risks going to and from your car in certain areas. Those things are real, and being able to minimize that is a really big deal. Okay, as promised, it is time for me to stop talking and for you to hear from our friend, Mr. Andy Zuvich. I'm going to let Shelly introduce him, and I'm going to go quiet for a bit.
Hi, everyone. Thanks for joining us today. I'm really happy and honored to introduce Andy. I know he's colleagues with quite a few of us here, but just in case for anyone that doesn't know him, he's a senior research scientist at Insight in Wilmington, Delaware. He has been a QLEX researcher for about 20 years, and that's been divided across 3 different institutions. So you can imagine there's quite a variety of study designs that he's tried with this equipment, which can range from your normal PK to bile, metabolite collection, IVGTT for metabolic syndrome, microdialysis, safety farm, first pass, and even working with mouse models, which is tricky in general, but especially with cannulated models. And last but not least, the novel experience that we're all having now is learning how to run animal studies in a pandemic. So he wanted to share his experience with QLEX and the efficiencies that it had already put in place made catching up after all of our time in quarantine much easier than even expected. So I'm going to turn it over to Andy, and just want to thank him for taking the time to chat with all of us. Thanks, Andy.
All right, there we go. Yeah, thanks, Shelly, for that introduction. Today, I just want to share my experience with the QLEX and how its efficiencies prepared us for the pandemic without even knowing it. So at Insight, we purchased our first 2 QLEX carts in 2015. And over the past 5 years, we have increased to 6 QLEX carts for a total of 24 stations. We have seen an increase in PK studies every year since getting the QLEX. In fact, we reached a high of a 66% increase from 2016 to 2017. And it's something I want to emphasize to like the bean counters. There was no change in head count during this time. It's been me and my coworker for the past 5 years doing the majority of the in-house rodent PK at Insight. Automated sampling has allowed us to increase productivity while maintaining quality samples with a small, experienced workforce. The flexibility of the QLEX and automated sampling has also allowed us to pursue other goals in both in vivo studies and administrative duties. Some examples are my coworker is the iCook compliance coordinator, and just last year was responsible for implementing a new web-based compliance software. I am one of the administrators for our tox data collection software, and I'm also actively involved in developing a CRO outreach program / assessment tool. We can do all this administrative work. We can help our fellow in vivo coworkers with tox studies, doing necropsies, all because our time isn't spent manually collecting samples. So needless to say, I'm a big fan of this technology for many reasons, but I think 2 stand out. And I can't just kind of touch on all these, but the point deserves to be repeated. First, less stress for the animals during sampling. There's no taking the animal in and out of their home cage for every time point. No restraint, no manipulation of cannulas or painful needle sticks. Usually the animals are unaware that blood samples are even being taken, and they get to stay in their home cage for the entirety of the study. That's a great refinement for our animal subjects. Secondly, and this is something I really love, it's the efficiency. It says it in the title. It's blood sampling. It's automated blood sampling. The Cool-X does all the work for you. And our equipment is very dependable. Our equipment runs from Monday to Friday every week. And our Cool-X is nearly 18 years old. And with a good maintenance plan, which we have, our equipment might run forever. So, in the rare case, one of our pieces of equipment needs repair, our maintenance plan and staff take care of us. So in the 5 years that we've been running numerous of Cool-X studies, we've never had any downtime for equipment. And you know what? Again, Candace mentioned this, but no more late time points. So for me, who lives, I like to think I live a somewhat busy social life before all this happened. That's worth the price of admission alone. No more staying late, no more going back during those cold winter nights at 7 and 10 o'clock. In fact, I haven't had to go back for a late night sample in almost 20 years, and that's a big refinement for the scientists performing the work. So, anything that makes our job easier, especially during these times, who wouldn't want to go for that? Another example of refinement for the animals, we use commercially available vascular access buttons for our rats, and eventually, hopefully, for our mouse PK studies. This allows us to reuse our animals for additional PK work. We dose our animals for PK sampling, and after the last time sample is taken, we flush and lock the vascular access button and return them to their holding room. We let these animals wash out and use them again the following week. So from the time the animals arrive to the time of their last study, excluding time they're on the Cool-X, these animals are housed together, which improves their quality of life. As a result of using these buttons, we have decreased our animal use by almost half over the past 2 to 3 years. Now, we like mentioning the 3 R's, and I mentioned 2 of them, reduction and refinement. So when following the principles of the 3 R's, I think it's rare to find a method that refines experiment to the benefit of the animal subject and reduces animal numbers, but it also makes the scientist more efficient and productive. A lot of times refinement can mean extra work for the scientist or the technician. So if there ever was a moment in time for the Cool-X to shine and utilize all these efficiencies, it's right now during the pandemic. So here's a little bit of our COVID timeline and insight in how we managed our PK studies during limited staff times. So in late March, we stopped almost all vivarium and lab activities. The campus was shut down with only essential personnel allowed to come in. Our order to work from home lasted until early May, when slowly we allowed a few other essential personnel that could return to work. With automated sampling in mind, we were among the first to go back to work. Now, we had the order to make sure we do these studies, but safely, to limit our time at the office and the desk, limit our time at limit our time in the vivarium, which will allow other vivarium users to come in and use that same space without unnecessary crowding. So once we received confirmation of the surgical models, we were able to start up where we left off with only very few small modifications to our schedule. Upon returning to campus, we had to wear the mask and the gloves throughout the building. In the vivarium, we had to wear full PPE. And to save money and inventory on the mask that we all wear, essential personnel were giving PAPRs. Think of like Darth Vader helmets, but more attractive. Very lightweight, minimal battery weight. The airflow lasted many hours. Very convenient. So we set up our animals the day before dosing. That way the animal can acclimate to the Cool-X caging during that time. So it's Amy and I doing these studies. One person would go in the Cool-X room and set up, the tubing sets, put the animals on, while the other person would complete paperwork, labeling the tubes, etc., in an adjacent lab. Now, during these studies, there was really only one time that my coworker and I worked together in the same room. So for the day of dosing, both workers were present to keep the time points tight and to be available for any troubleshooting, whatever that might be. After dosing, one person would leave to work the remainder of the day from home, while the one person would stay through the 4-hour time point. During that time, they would do observations, process samples, and we would feed the animals after the 4-hour time point. At that point, the second person, which was usually me, I'd finish my day working from home. The other person would come back to process the later blood samples and the urine samples. At the 24-hour time point, again, working in separate rooms, One person would break down the Cool-X, while the other person would start the ball rolling on the following day's studies, and we would repeat as necessary. Again, especially during coming back from COVID, we were running the Cool-X Monday through Friday. So during the height of the pandemic in Delaware, we followed, company orders by limiting our time at work just to over about 4 hours a day at most. Our exposure to each other during the Cool-X studies was no more than 30 minutes per day. That's with also full PPE. So everybody has a schedule to keep up to, and they asked the same of us. So we were able to catch up with the studies that were canceled in April and early May, and still continue the current schedule, and we caught everything up by the end of June. So we caught up basically with 6 weeks of backlog studies in no time. The big thanks to our Cool-X and flexible staff. Currently, we're kind of back to things as they were. We still kind of are able to work from home, but without the Cool-X and having to do manual samples, exposure to each other would be very difficult. But the Cool-X allows us to get our work done, be more productive, and work safely during these COVID times. That's it for me, Candace and Shelley. Thank you for listening.
Thanks, Andy.
Yeah, no problem.
So that's it for us today, except we are more than happy to answer any questions that you have. I don't see any in the chat window. I do see that a lot of people were putting in there that the video wasn't working, so I'm very sorry that I didn't see that, but we will make sure to send a link to everybody who registered for this. And we'll make sure that you get access to that. So we'll give it a moment here to see if anybody has any questions they want to put into the chat window. And if so, either Andy or I will be happy to answer those. While we do that, I would invite you to visit our website. You can see that here, basi-coolx.com. You'll be able to see the video there as well if you wanted to go check it out. There's also a really nice bibliography search page, so you can look at publications using the Cool-X under a variety of circumstances for the small and large animal models, and learn a little bit more about how people actually put it to use. And then just a reminder that the ALAS webinars, there will be no webinars next week because we will be all attending and having a great time at the virtual ALAS meeting. The series will resume on November 5th, and you can see the updates on the ACE community and also the ALAS LinkedIn site and ALAS site for additional announcements. I do have a question here. Let's see, this one's for Andy specifically. Nobody wants to hear from me. Andy, are all of your early discovery studies run on the Cool-X?
For PK, the majority of them are. We're mostly a mouse facility. But as far as our PK goes, yeah, I mean, majority of the studies, all the rat studies are done on the Cool-X.
Okay. And are there any study designs besides the ones you just mentioned in terms of the mice for some of them? Are there any study designs that you've had to do manually, or could you maybe expand on like which is a good fit?
Well, for rat, we do it all on the Cool-X now.
Mm-hmm.
The mouse, we're still trying to hammer those details out. We're also using buttons with the mouse studies, which are working out really well, which allows us to use them on the Cool-X a little better. But one of the biggest, I think, time constraints previously was doing the bio studies manually with blood sampling. That was quite a chore. That had to bring in quite a few people for that study. But now that we have we automate the blood sampling with the bio studies, we get that done pretty quickly now. So that's been a nice addition.
Great, thank you. I've got one more here, which is somebody would like to know, were there hurdles with IACUC approval when you brought in the new technology?
Well, no. And then one of the reasons for that is some of the staff that was at Insight at the time was very familiar with it. We did some comparison studies. for DMB, you know, comparing manual to Colex, and it all worked out. But most of the iCook staff, or a lot of the iCook staff, I would say, was very familiar with the Colex before it was brought in.
Right. I'm going to answer that one a little bit too, just because you have a sort of unusual circumstance there that a lot of your folks knew it. So we do have that as a challenge sometimes. And really, once the staff understand the difference between what the system can do, it gets a lot easier. But that is sometimes a question that comes up, or that we have to support our customers with. So when you do run into those issues as new customers, we're happy to help. We have, it's a little bit different because we know the technology too, but our company is both a products company and a CRO. So we have our own lab where we run studies on the Colex, and we have an iCook there. And so we know what it's like to have to explain the use of the system and justify the use of the system, which makes it a little bit easier for us to support customers that are dealing with that same thing. In some places, it's no problem at all. In other places, it's really just a matter of educating about the technology so they understand what's different and how that works differently for the team. All right. Well, I don't see any other questions popping in here, so I will close it up with an additional thank you very much to you, Andy. We appreciate you joining us and sharing your experience. It's very helpful for people to understand how you could put it to use, so thanks.
You're welcome.
And thanks to all of you for joining us today. Sorry about the technical difficulties on the front end. 2020, yes, and for the video being frozen, so we'll make sure to get everybody a link to that. So thank you all very much for your time. We really appreciate it, and hope you have a great rest of your day.